My name is Martha Crago, and I'm the Vice Principal of Research and Innovation at McGill, and it's my pleasure to be here with you tonight. I'm delighted to be here for this, what I think is going to be a very exciting evening, that's going to highlight the kinds of brain research that's done at the Ludmer Centre for Neuroinformatics and Mental Health. And I want to mention that this is being live streamed, so welcome to our online audience as well, which means there's a lot of people out here tonight, and that's exactly what it should be. So there's a packed evening for you. I hope you've enjoyed the science fair portion and heard from the young researchers that are at the Ludmer Centre researchers about the kinds of research that they're doing. I found it very exciting to hear from the three that I had time to visit. So we wouldn't be here tonight if it wasn't for somebody's great vision. And that great vision came along with generosity. And that was the vision and generosity of one of McGill's most notable alumni, Mr. Irving Ludmer. And last spring, Mr. Ludmer and the Ludmer Family Foundation made an extraordinary gift of $10 million to support brain research at the Ludmer Centre. This means that the Foundation's gift in neuroscience at McGill and its partner institutions is $18 million. This is simply extraordinary. So, chaque année, les maladies mentaux touchent environ une personne sur 5 au Canada. C'est pas petit. Certaines maladies comme l'autisme touchent les enfants, d'autres comme les maladies d'Alzheimer et Parkinson touchent les personnes âgées, et encore d'autres, comme la dépression, les troubles anxieux, peuvent survenir à tout âge. So we have a big task ahead of us, which is to help figure out how this works and what are the best things that can be done about it. So Mr. Ludmer's vision for advancing discoveries in brain search is very much aligned to another very generous McGill donor, Mr. Larry Tannenbaum, who is with us here tonight, and whose transformative $20 million gift established the Tannenbaum Open Science Institute at the Montreal Neurological Institute. But before I go any further and we get into the heartland of tonight's event, I want to introduce McGill University's principal and Vice Chancellor, Suzanne Fortier. So Professor Fortier is a native of Quebec. She graduated from McGill with a BSc and a PhD in chemistry. Elle exerce ses fonctions de principale et vice-chancelier de l'Université McGill depuis septembre 2013. Et avant d'être nommé principal, le professeur Fortier a été président du Conseil des recherches en sciences naturelles et en génie de Canada. In 2017, she was appointed the vice chair of the World Economic Forum's Global Universities Leader Forum. And this year, in 2018, she was named an Officer of the Order of Canada. Mesdames et Messieurs, la principale et vice-chancelier de McGill.
Merci Martha et bonsoir à vous tous. C'est fantastique de voir cette salle remplie de gens. Nous sommes réunis pour deux choses. Une célébration de Irving Ludmer et de sa famille, de la fondation de sa famille, et aussi parce qu'on a tous le désir et le besoin d'apprendre sur le cerveau humain. There's nothing more precious than the human brain. And I know that that also brings us here together this evening because we all want to learn about this. Before I say anything else, I want to add my words of thanks to Irving. It's hard to call you Irving Ludmer. You're such a great friend. I always call you Irving. But to Irving and to the Irving Ludmer Family Foundation for their great support and their continuing support to research on the brain. His most recent gift is a very inspiring investment in the groundbreaking research that will be done at the Ludmer Centre. And believe me, Irving will insist that it is groundbreaking research. I'm looking at some of the researchers here because they know that that is absolutely true. It's also a great vote of confidence in the research that has been going on in the center, not in a very long time, and is already paying dividends. So it's great, Irving, and merci beaucoup. La vision et le but du centre Ludmer sont de faire une différence, une véritable différence, et de changer la vie de nombreux patients et de leurs familles qui souffrent de maladies mentales. C'est une maladie qui est présente partout, chez nous, à côté de chez nous, dans nos familles, chez nos voisins, à travers le pays, à travers le monde. Donc, c'est une vision qui est non seulement importante, mais qui est urgente et nécessaire. The fellowships and awards that are a fabric of Irving's ongoing support will be opening doors for a new generation of researchers and will provide them with great opportunities in education, in training, and it will equip them with the kind of cutting-edge technologies, big data, and all of this to work in advancing our knowledge of the brain, and all of this in an open science model. Marta mentioned the gift from Mr. Tannenbaum. What is great here, I'll tell you, is that both Irving and Larry understand well what is needed to make progress in this area. We need to work together across the world in sharing the knowledge, in sharing the discoveries. Progress is necessary, it is urgent, and it will only happen if we work together and we are really, I think, aligned with your vision, and we're very happy that you're supporting that. J'aimerais aussi souligner qu'il y a beaucoup de gens ici qui appuient cette recherche sur le cerveau et qui partagent notre vision. Alors, j'aimerais vous remercier, vous aussi. Collectively, you help us in providing the very necessary funding so that the research can be carried and a research that is needed for society. Alors, je vous dis merci. Thank you very much. Et maintenant, j'ai le grand plaisir, it is my great pleasure, to call on Irving Ludmer, to say a few words. Now, I'll tell you a few things about Irving just before he gets to the stage here. He graduated from McGill with a Bachelor of Engineering in 1957. He was an executive with the Steinberg grocery chain for many years, and he's a founding partner and president of the investment company Kleeman Ludmer Steinberg. In 1989, he established the Irving Ludmer Family Foundation, and his foundation supports very much needed areas: social services, the arts, hospitals, health agencies, education and initiative, and of course, research. Irving was a long-serving member of the Montreal Neurological Institute and Hospital Advisory Board, and through the Ludmer Family Foundation is a longtime supporter of McGill and of brain research. He contributed also to the renovation of the Department of Psychiatry's Ludmer Research and Training Building. And in 2013, he became the founding patron of the Ludmer Centre. Irving never shies away from big challenge. You know, when he came to McGill, he enrolled in engineering physics because this was the hardest program that you could enroll in, and I can tell you that this is still true today. And so when he chose brain research as an area to support and to stimulate, it was fueled by the same spirit. This is one of the toughest areas of research, and he's not one who's going to shy away from it, and that's inspiring all of us, and particularly our researchers, to pursue their great work in this area. Irving is a community leader. He's demonstrated both the most complicated problems of that our society face and the most innovative research deserve resources. Alors maintenant, Irving, je vous invite à prendre parole.
Merci, Madame le Principe. I would like to thank everybody for coming out tonight. You're going to hear some interesting scientists speak about brain research, and it's quite the challenge. And these people are up to it, and I hope that over the next number of years, we'll see some major breakthroughs. So you've heard about Larry Tannenbaum's Open Science Initiative. And I'd like to thank Larry and tell him that I think I told him once already that I knew his dad very well and tried to buy land from his dad in Ontario. And his father took me to a restaurant on Bathurst Avenue where he said, I have a guest, so we're going to each have 2 hot dogs and something to drink. And it turned out that a hot dog was a pastrami sandwich of major proportions. And I don't remember the name of the restaurant. I do remember one thing, that after I tried to eat the second sandwich, I sat down in the front of his father's Cadillac, and I was just praying that we get to wherever we were going before I regurgitated the whole goddamn thing. But he was quite the character, and built a beautiful business, and obviously a family that cares. So one of the things McGill's neurological research is famous for is now Larry's Open Science Consortium. And it's extremely important because if everybody's going to repeat, Einstein once said, if you do the same experiment over and over again and it keeps failing and you keep doing it, then you got a mental illness. And so here with Open Science, people from everywhere that are associated with the group can contribute. Everybody gets the same results, and the amount of duplication is bound to decrease. And from there, you're going to get much faster progress and much better progress if everybody's working together. So that's one of the big things that McGill has done. And another is the CFREF thing. I don't know if you're familiar with this, but there was the federal government put on a sort of a competition called the CFREF, and McGill out of that won, I think, some $84 million spread out over a number of years. I think, was it 7 years, Suzanne? Yeah, 7 years. And the University of Montreal got about 90 some odd million dollars. And Western University also got a very nice stipend, which I have written down here, at about 60 some odd million. So these 3 universities are now not just spending the government's money, but cooperating in research, in doing what they're doing. And now for the big dream, once we're all together, is to make a global consortium. And already, Alan Evans and his team, Dr. Alan Evans, sorry, Alan Evans to me, Dr. Alan Evans by real statute, and they have already signed an agreement with both Cuba and China for various research projects. And now there's an advisory committee that is suggesting whom should join in a global consortium, doing different things, approaching the problem from different angles. You know, you have expressions like you are what you eat. And that's what the brain is fed through metabolic processes, and it's extremely important. They used to say, I have a gut feeling. The gut feeling has now become the microbiome, with trillions of bacteria that live within us. And I could go on in epigenetics and genetics and so on. But I'll save you that. Let the scientists know what who know what they're talking about, tell you about all that. So we look forward now to a global consortium with McGill at its heart. And I think we have the talent, the people, and a long history in neuroscience research. So let's take it from here. Let's see where we go. Thank you very much.
I'm going to pause a moment to share a teaching, if you will, that Irving Ludmer gave me. He said, in any room, if you ask, who here does not have a member of a family that has a mental illness of some kind, you will see very few hands go up. And I think that shows you how, I think of that often, And I think it shows how penetrating your gift and your generosity is. It affects us all, frankly. And now we're going to have the fun at looking at how that generosity is accelerating discoveries forward in brain science. So I'm going to introduce 5 people to you, and then they're going to get up one at a time and talk to you for 5 minutes, although one of them says it's going to be 6. And they've got this huge clock here to remind them of every second that's going by. But I'm not going to be watching, I'm not going to be ringing bells, I'm not going to be doing any of that. They're self-monitoring. So they will be giving us these 5 talks, one after the other, and then following that we'll be able to have a good question and answer period with them that I hope you'll participate in. So the first person who will speak is Rosemary Baggett, who is an assistant professor in McGill's Department of Psychology and a William Dawson Scholar. She leads a team of researchers looking at the mechanisms of depression, and tonight she'll speak on how stress shapes the brain. Following her will be Dr. Kieran O'Donnell, who is an assistant professor in epigenetics and epidemiology in McGill's Department of Psychiatry, and an Sifar Israeli global scholar in child and brain development. His work at the Douglas Hospital Research Centre examines how early environment shapes the child development to identify those at risk for adverse mental health outcomes. And tonight he'll speak to us about why maternal mental health matters. Following him will be Sharif Karama, who is an assistant professor in the Department of Psychiatry and also works at the Douglas Hospital Research Centre. His research is focused on the impact that genes, environment, and their interaction have on brain development and cognitive ability, and he will address IQ in the brain across our lifespan, and he's the one who's taking 6 minutes. And then he will be followed by Dr. Celia Greenwood, who's co-director of the Ludmer Centre, an expert in quantitative life science statistics, and a senior investigator at the Jewish General Hospital's Lady Davis Institute. At McGill, she's a professor in the Gerald Brofman Department of Oncology and a graduate program director in the Quantitative Life Sciences PhD program. She'll speak to us about transdisciplinary big data researchers, tomorrow's innovators. And finally, Dr. Alan Evans is co-director of the Ludmer Centre at the Neurological Institute and a pioneer of neuroimaging techniques and important biomarkers for the early diagnosis of neurological and psychiatric diseases. He's a James McGill Professor of Neurology and Neurosurgery and the holder of the Victor Dadelaide Chair in Neuroscience. His talk will be called Towards a Global Brain Consortium. So he will leave us off where you brought us to. And that will be the final presentation, and then we'll have our questions and answers. So welcome, Rose Baggett.
50 years ago, my grandfather died of a heart attack. One evening, he was feeling unwell, and he went to bed early. The next morning, he was dead. A few years ago, my father was feeling unwell. For a while, he'd been complaining of low energy and some aches and pains. His wife pushed him to see a doctor. And in a simple operation, he had a stent put in, and today he's visibly reinvigorated. The incredible progress that we have made in recognizing and treating heart disease in just one generation is an incredible, powerful success story of modern medicine. Scientific research has developed treatments that can reverse the course of this disorder. But in the same period, we've seen very little progress in psychiatric illnesses, which impact the lives of 1 in 3 Canadians. We know that 1 in 10 Canadians report experiencing mood or anxiety disorders. We also know that these disorders are more common in women than men. And of those affected, more than a third report that they're unable to work at some point in time because of their disorder. The World Health Organization has identified one single psychiatric disorder as the leading cause of disability worldwide. and that is depression. People who experience depression will often find that it reoccurs throughout their life. And many people don't find current treatments to be effective. When they do work, they work by suppressing symptoms, not by treating the underlying cause. One of the biggest challenges we have in developing more effective treatments for these disorders is that we don't really understand what causes these illnesses. We don't know what's going wrong in the brain. And we don't have a biological test that could confirm a diagnosis. We do know that stress is a major risk factor for psychiatric illness, especially for anxiety and depression. Stress floods our brains with the stress hormone cortisol, and over time, this can lead to permanent changes in brain structure and function. In my lab, we study mouse models to understand how stress is changing the brain. From these experiments, we've already learned that stress is changing the way that brain cells talk to each other. It's also changing how brain regions communicate across networks. And we've started to discover how stress is more permanently shaping the conversation in the brain by regulating the activity of genes. So we can think of stress as being a bit like a dimmer switch that's increasing or decreasing the activity of groups of genes. And RNA sequencing is a technology that we can use to measure the activity of genes across the entire genome. So by doing this to profile gene activity in multiple brain regions in many animals, we can amass large amounts of data that can help us identify patterns in how stress is affecting the brain. We can also identify key regulators that control these groups of genes that are being impacted by stress. We can then use this knowledge to develop new drugs to target these changes. But we also know that not everyone reacts to stress in the same way. So I think you probably all know someone who, no matter what life throws at them, they continue to navigate life's challenges with incredible resilience. We see this in humans, but we also see this in our mice. We can study the brains of these resilient animals, these resilient mice, to understand what's different in their brain that protects them from the effects of stress. And we can use this to develop treatments that might build resilience in the face of stress. Another really important challenge that we're working on is trying to understand and predict what makes an individual become resilient or susceptible to future stress. So we're trying to understand this, to understand how stress interacts with the brain. By studying the brain before stress, we can get a clearer understanding of what the underlying mechanisms are in these stress-related disorders like depression. And so this can lead to treatments that might be able to reverse the effects of stress, the damaging effects of stress in the brain. But really importantly, we might be able to develop interventions that can prevent the deleterious effects of stress before they ever occur and prevent disorders like depression from ever emerging. So big data and new tools in neuroscience have really brought us to a tipping point. By using data as our guide, we can generate fundamentally new ideas about how the brain works. And we can test these in the lab using these cutting-edge arsenals of tools. With the help of Miguel and the Canadian Foundation of Innovation, I have equipped my lab with some of the most advanced techniques in neuroscience. And my incredible team of young researchers are applying these tools to generate unprecedented insights. We are visualizing, but also controlling the activity of individual brain cells while animals are interacting with their environment. We're also starting to probe how experience rewires the connection between brain cells. As well as measuring the activity of genes, we can control the activity of genes to really understand how they shape the brain. By harnessing the power of big data, We can change the face of depression in this generation.
Good evening. Full disclosure, this may also be 6 minutes. So tonight we're talking about big data. Big data is fundamentally revolutionizing how we think about the human brain in development and also in disease. Big data is fascinating, it's exciting, but sometimes simple data, relatively small numbers, can also have an impact. They can have such an impact that someone like me dedicates their research career to understanding them and to trying to do something about them. So there's some relatively simple, small numbers I'd like to share with you tonight that I'd challenge you to take away, if nothing else from the next 5 minutes, just take away these simple numbers. And these simple numbers are 1 in 5 and 8. So 1 in 5 and 8. So what are these simple numbers? What do they represent? So 1 in 5 represents the number of women who will experience a perinatal mental health problem, most commonly anxiety and depression. And what we know is that not all women are affected equally. There's other women in high-risk groups like Aboriginal communities, in low and middle income countries that show significantly elevated rates of perinatal mental health problems, as many as 1 in 3. A third of women suffering and struggling with their mental health problems. And we know that around 50% of these women that show symptoms in the postnatal period actually show symptoms in the prenatal period as well. Antenatal anxiety and depression is just as common as postpartum or postnatal anxiety or depression. Here in Quebec, that equates to roughly around 20,000 women per year. And sadly, here in Quebec right now, we don't actually ask women how they're doing in pregnancy. We have no coherent strategy in Quebec, but also nationally, for screening for antenatal anxiety and depression, and that's something that I'd like to change. Of course, once you do that, then you have to make sure that the resources are in place to help these women that you've identified, but that's a conversation for another time. So 1 in 5. What about the 8? Well, that's a number that drives my research forward because that's the cost of perinatal mental health problems in the UK, £8 billion per year, per annual cohort of live births. And what I find just striking and startling about that number is that 72% of those costs are derived from the effects on the child. So what effects on the child? Well, here's some data from my own work in a cohort of over 15,000 pregnant women with outcome data on around 8,000 children from 4 to 13 years of age. But I could have taken this figure from multiple cohorts around the world that illustrate that maternal mental health in pregnancy and in the postnatal period has a significant and marked effect on child mental health. So what you see in the upper part of the graph are children born to women that experience anxiety in pregnancy and in the postnatal period. And you see that they have the highest symptoms of emotional and behavioral difficulties that are evident at age 4, that persist until at least age 13. We've done follow-up analyses to adulthood until the mid-20s, and we still see these effects. Now, while this might be a depressing graph, I see some hope in it, because I think what you can appreciate from this graph is that if you are exposed to anxiety at one or another time, or if you're not exposed to anxiety at any time point, well, then you do better. You have lower symptoms of emotional and behavioral difficulties. And I think that's promising because we know that there's interventions that we can implement right now that will reduce anxiety and depression. The other thing that you might not see from this graph is that not all children are affected. In fact, children can be affected in very different ways. And through the help of the generous donation from Mr. Ludmer and his foundation, within the Ludmer Centre, we're focusing on trying to understand why some children are affected and others are not. Why does one child end up with higher symptoms of inattention and hyperactivity, whereas another child might end up with higher symptoms of emotional problems? At the moment, we have no way of accurately predicting that. Which child is going to have persisting symptoms through adolescence and into adulthood? Again, we can't predict that. And what we're doing is focusing on the building blocks of life genetic variants and how they function and how they're regulated, looking at chemical modifications that are placed onto the DNA, so-called epigenetics. And we're integrating information on sometimes millions of variants, up to 19 million genetic variants, with information on function, sometimes up to 850,000 sites where we can see these chemical marks, to try and better understand who is at risk and who will benefit from interventions. We're doing this in a global context. We're partnering with collaborations, early interventions in the United States, where we provided the first evidence just this year that an early intervention program influenced these chemical marks on our DNA. We showed this 25 years post-intervention, and now what we want to do is see when exactly this occurs. Is this evident at birth? And to do that, we're collaborating with colleagues at Warwick University and at Oxford University, who are leading an evaluation of a £160 million investment of early intervention programs in the UK. And we're collaborating with them to assess genetic variation and epigenetic variation. We're not forgetting the developing world. We're collaborating with colleagues in Vietnam who are implementing a randomized controlled trial of a perinatal intervention that seeks to reduce anxiety and depression in women in rural Vietnam. As you'll remember, as many as 1 in 3 women in rural Vietnam will experience significant perinatal mental health problems. So I started my talk with the title, Why Maternal Mental Health Matters. Well, it matters not just because of the human cost, but also the economic cost, the effects on the child. And I think there's been no better time in science for us to be able to use big data to understand how we can improve interventions to ensure that they work for all and not just for some. Thank you.
Good evening. If I give you all an IQ test right now, and I order the scores from highest to lowest, and then gave you another IQ test, completely different from the first one, and ranked the scores again from highest to lowest, well, the high scores in the first test will tend to be the high scores on the second test. And unfortunately, that's the case for the low scorers too. So I don't know about you, but I find this shocking and a little bit unpalatable. But, ladies and gentlemen, this is the most robust and reproduced finding in all of psychology, and got me intrigued. The other thing that I know is that both genes and environment influence brain development and can have an impact on cognition, cognitive ability, and IQ. An example for environment is that at the end of summer, children tend to have a decrease in their IQ. They've been out of school for 2 months. Now, I'm going to give you a metaphor that kind of ties all of these things together. Imagine a circular elastic. It's a brain. And you can stretch this elastic. Some people have an elastic that is easier to stretch than others. And the way to stretch the elastic is with environmental stimulation, intellectual stimulation. IQ does not measure how easy it is to stretch your elastic, which is more something that comes from our genetic background probably. What IQ measures is the area of the circle, and it can move to some degree as a function of our intellectual stimulation. Keep that in mind because it's going to give you some perspective on how to understand some of the things I'm going to share with you tonight. So, as part of Ludmer Centre scientist, I was granted access to large data sets. One of them is the NIH-funded MRI study of normal brain development. These are children aged from 4 to 22 that came to the NIH every 2 years, had an IQ test and a brain MRI. The second cohort is the Lothian birth cohort of 1936. So picture this. We're in 1947, and the Scottish government decides to IQ test all 11-year-olds in the country. And the reason they did that is that they thought that their children were getting dumber. But it didn't pan out. They were not. Now, the beauty of this data set is that in their 70s, the survivors that were in Edinburgh were retested. And at ages 73, 76, and 79 had an MRI. Now, these data sets and others allowed us to find the following. The greater the volume of your brain is, the higher IQ tends to be. The greater the thickness of your cortex is, which is the outer layer of your brain that is crucial for thinking, the greater your IQ tends to be. The greater the quality of your myelin, which is the sheath that surrounds the wiring in your brain, the greater your IQ tends to be. This sheath allows for more efficient and fast processing and allow good network efficiency across your brain. Now, these make somewhat independent contributions, and we're getting closer, and there are more findings that I'm not discussing here, but we're getting closer to being able to actually estimate IQ on the basis of a brain scan. And I know this is scary. It scares me too. Now, IQ tends to be stable, but it can change dramatically. And in the NIH, within a 2-year period, we've seen children having an average IQ, and after 2 years, ending up with an IQ in the top 5% of the population. Another interesting finding is that IQ at 11 predicts brain structure 60 years down the road. 60. Now, this might give you the impression that things are fixed. They are not necessarily fixed. We can have an impact. So a big question is, how can we increase our chances of maintaining our intellect in old age? So remember, we have IQ at 11, we have IQ at 70, 73, 76, 79, and we know what happened in between in terms of life habits. So this is what we found. People that exercise regularly tend to stay sharp in later life, or increase their chances of staying sharp later in life. People that get regular medical checkups that could detect diabetes or high blood pressure also tend to increase their chance of remaining sharp later on in life. Eating healthy, Mediterranean-type diet, nuts, olive oil, limiting in red meats, also tend to have higher levels of cognitive ability later on. Sleep. People that sleep 7 to 8 hours a night are those that increase their chances of staying sharp later on. We tend to underestimate the impact of sleep. Don't smoke. We've noticed that the more you smoke, the more your cortex thins. This is somewhat reversible, but it's a very slow process. I had people that had been smoking for 20 years or so, 20 cigarettes a day. It took them roughly 20 years to catch up to the level of their age-matched peers. So it's a slow process. The earlier you stop, the better it is. Take up a new activity, like learning a new language, dance, exercise your brain in general. And something we should not underestimate is social connection. It's really surprising that there's a very strong link between being connected and talking to people, having people close to you, and longevity and maintaining your cognitive health later on in life. So my message to you is, it's never too late to start, but the earlier you do, the better it is. It's a way of life. There you go.
As you may have seen in the media, and as you've certainly heard already from the past 3 speakers, there's a data deluge out there. All fields of research are being transformed by the massive amount of data that is being generated, and many different types of data as well. But this data deluge, this transformation, or perhaps I should even say this data tsunami, creates a huge potential. So we can learn from this data We can understand what is going on in the development of the brain. We can understand what is going on as illness comes along. But this potential is only going to be realized if we have methods to analyze the data and software packages to analyze the data and knowledge in how to do that appropriately. So these are my two areas of interest. My research group works on the development of methods and software packages that go along with that for the analysis of big data. I have a group that has been generously supported by the Ludmer Centre and the Ludmer Foundation to work on many aspects of developing new methods in this context, trying to increase the capacity to find gene-environment interactions, for example. I want to give you one concrete example. I have a collaborator at Saint-Justin, Dr. Jacquemont, who works on the effects of copy number changes in the genome. So these are small sections of DNA that are lost or gained. And children come to the clinic and they want to be able to predict what is the future cognitive ability and intelligence of these children who are affected by these copy number changes. But they are extremely rare. And so you cannot look at other patients to see how other patients will do. So I've been working with this group for a couple of years, and we've been working on trying to develop methods to do this prediction. Along the way, we have developed a software package that develops a kind of model called a kernel model. A kernel model is a kind of machine learning method that takes in data of various kinds, as you can see here, and puts it into a kind of a black box and spits out a prediction at the other end. A lot of black box models are really hard to interpret, but the software package that we've been building allows you to have an intuitive visualization of the contributions of the variables that go in. So as I said, this is just one example. I could say that we're trying to break open the black boxes. I have collaborations with many of the Ludmer Centre researchers, and so does my group, and we're working on methodology developments in many different areas and contexts. The other area that I'm really interested in is how do you develop the researchers of the future? And what is the educational aspects there? So you could think of the educational aspects in a couple of ways. You can imagine in the blue arrow taking people who have a strong quantitative formation in mathematics or physics or statistics or computer science, who wish to then improve their education and their knowledge in a life sciences or a neurosciences domain. So McGill has just launched a PhD program that does exactly this. It's called Quantitative Life Sciences. It was mentioned earlier. And I'm the graduate program director of that at this time. One could also imagine a different kind of flavor for training where you take people who have been working for many years in a neurosciences domain or in another life sciences area and have deep knowledge there, but are now realizing that they are being inundated with data and they would like to improve their data analysis skills. So again, McGill has a lot of initiatives underway trying to do this. One of those is the McGill Initiative in Computational Medicine, which is trying to design some new educational opportunities. And I'm sitting on the education subcommittee, chairing the education subcommittee in that endeavor. I would also like to highlight the Ludmer-sponsored research training workshops, which happen periodically several times a year. I take a lead role in organizing those, where we invite speakers and organize workshops around them to encourage interdisciplinary collaborations and discussions. So what is big data? Big data is not very useful to us unless we can use it to do inference. And inference is the idea that we don't just want to have the conclusions of the data that's sitting in front of us. We want to be able to extract a general principle that will hold true tomorrow. In order to do good inference, well, we need methods, we need software, and we need understanding. These things together will transform the data deluge into a manageable information flow and create tools for tomorrow's innovators.
Good evening, everybody. I think they had me batting cleanup because I couldn't be trusted to stick to 5 minutes. This is a very exciting time to be involved in neuroscience. We're in the middle of an information revolution that's affecting all of our lives in many different ways, and the same is true of all science, but particularly neuroscience. We are swamped by a vast amount of data. You've heard tonight people talking about genes at one end, subcellular levels, cognition at the bottom end, and everything in between. There's a problem. We're overloaded. What do we do with all of this data? And bear in mind that it's data, it's not knowledge, and it's certainly not wisdom. To convert data into knowledge, we need a vast amount of analytic capabilities to extract that information that informs us about brain disease, brain disorders, normal development, brain plasticity. And we're in the midst of bringing together all of these different disciplines to allow us to extract that information from the data. You will have been struck, I think, by the youth of the people that have been speaking before me. That's not an accident. You have young people coming into neuroscience who have very, very broad training in mathematics and physics or neuroscience or psychiatry, neurology, and somehow we have to get all of these people to talk to each other. And this is both an enormous challenge and something that is extremely exciting to watch the new ideas which are emerging from these transdisciplinary interactions, which are more and more common. We can think of neuroscience at many different levels. We can think of it at the molecular level, all the way through to the clinical level. Increasingly, we are thinking about the brain not as the site of brain disorders where you might have a stroke or a tumor, some focal disorder, but as a distributed entity where disorders cannot be identified and localized in any particular area. They are a consequence of disorders of wiring and connectivity in the brain. And many of the mental disorders that we are considering are being understood at that level, aberrations in connectivity. To pull all of this information together, we have to operate, we have to introduce neuroinformatics, which is essentially what those little green boxes there show, databases, tools, and models. This is the framework wherein we can draw all this information across different scales into one common framework. And I'll give you one example of this that is really the work of a young man who's in my lab for the last 4 years as a postdoctoral fellow, Yasser Aturi Medina. Yasser is now a grown-up. The most important thing I've done in the last few years is get Yasser accepted as a faculty member at McGill University. We are very lucky to have people of his stature to take the future forward for us. And the particular example that I'm going to talk about there is that we can define the disease as a propagation throughout that network, Alzheimer's disease in particular. And when we can model that disorder propagating through time, and we can start to design interventions which would interfere in the mechanisms of that brain disorder. It's no good just looking at the sequela, the outcomes of this brain disorder. You have to understand the mechanisms that give rise to that disorder. That's what big data allows us to do. At McGill, we've built a large amount of computational infrastructure to provide the basis for everything that I've just described. You see here tools which allow us to store multi-domain data together, to conduct high-performance computing analytics of those data, and to visualize the results. Now, all this machinery is great, but it has to have an endpoint, and the endpoint is a better understanding of Alzheimer's disease, a better understanding of Parkinson's disease, a better understanding of autism. The big questions now are, can we make all of this data available to our colleagues around the world so that more people can take that data and ask questions of it and extract information from it? The days of one scientist, one lab, one paper per year are moving away. Increasingly, data is being made available in an open context And lots of people can get information out of that, which ultimately expedites and speeds our arrival at a cure. That's why we're doing this, not for the joy of the science itself. It's ultimately to come up with cures. So we are increasingly engaged with our colleagues around the world. These are many of the groups that are working with the Ludmer Centre scientists as we speak. Many people around the world. I'll just give you a couple of brief examples. The Quebec Parkinson's Disease Network is using this infrastructure that I've described. The US study of autism. The Canadian Network for Consortium on Neurodegeneration in Aging, looking at Alzheimer's disease and other forms of dementia. The Ludmer Centre really is a binding principle that allows us to attack all of these disorders with a common set of tools and analytic capabilities. The Ludmer Centre gave rise to the Healthy Brains for Healthy Lives initiative that you've heard about from Irving Ludmer. This was another recognition of the importance of big data applied to brain science. It's also been the basis of the Canadian Open Neuroscience Platform with support from Brain Canada and from the Tannenbaum Open Science Initiative. Larry Tannenbaum's in the audience, and Irving Ludmer. So all of these are ultimately larger and larger and larger Russian dolls as we go out and out to further afield to ask bigger questions, to pull in bigger amounts of data from different countries. We're integrated with the Human Brain Project in Europe, which is a 1.2 billion euro initiative. We're working with our colleagues in China and Cuba. I could talk for hours about this particular initiative, but simply to say the principal form of dementia in the West is Alzheimer's disease. In China, it's vascular dementia. We don't know why. It could be genetics, it could be lifestyle, it could be nutrition. All of these reasons we need to understand better the mechanistic basis of these disorders. So we're going to bring all of these tools together. We're in the early stages of building a global brain consortium. We brought together leaders of many of these international initiatives to McGill in October last month, and we will be holding a plenary meeting of about 50 international scientists next year to consider these questions and build together a truly global assault on brain disorders and the normal brain development. Thank you very much.
Well, while you seat yourselves, let me say how remarkable it is to see the strong history of brain research that took place at McGill University and the Montreal Neurological Institute carried forward through this generation of researchers. So it's a real pleasure to have you. All right, now I'm going to ask you some questions, and I'll start with one that was written here for me, and I think Dr. Baggett touched briefly on this kind of issue, but I'm just curious, if you went through the line here, what would you say? It has been said that our understanding of brain diseases today is similar to what we knew about cancer in the 16th century. Do you agree, and why? Who wants to jump in?
I'm going to allow someone else to tell us what we knew about cancer in the 16th century. But I'm going to... you're right, but is there a switch or...
There we
okay.
go.
Can you hear me now?
Yeah.
Right, so I'm going to conclude we didn't know a lot about cancer in the 16th century. I think we know maybe a little bit more about psychiatric illnesses and other brain diseases, but our knowledge is rudimentary, and I think it's really interesting to note the ways that we've come about this knowledge. For instance, in the case of depression, which I know about, most research has focused on the serotonin hypothesis of depression. And the reason why we focused on that is because through serendipity, we found that there were drugs that were initially used for something else that acted on serotonin. And so because of this sort of happy accident, we decided depression must be all about serotonin. But we know that's not the case, because although some people do see benefits with SSRIs that target the serotonin system, many people don't. So I think we need to think about how we find this knowledge.
Somebody else want to jump in on this question?
Yeah, well, I'm going to be a bit provocative, and I'm going to say that the folks in cancer have an easier time of it. Partly, and that's not to negate the seriousness of it, but a neurologist colleague of mine was talking about brain cancer and tumorigenesis, and they say, well, we can identify the diseased tissue, and we can also identify the healthy tissue. And we can compare and contrast mutational signatures and different genetic aberrations in this tumor that can really give us an insight into the etiology, into the pathology. And my neurologist colleague said, in depression, in mental health problems, where's the tumor? Where's the diseased tissue? And I think what we're beginning to realize more and more through these really remarkable network-based analyses is that there's many genes, there's many networks that contribute to mental health phenotypes. And I think we may be lagging behind cancer a little bit, but I think we're catching up fast.
Somebody else wanted?
Well, I think in the 16th century, people didn't have an appreciation how cancer spread, so how there was communication between different parts of between the tumor and other things. But as Kieran said, now it's possible to actually get samples from the tumors, to actually analyze them at a single cell level. Very few people object to having their tumor removed. But on the other hand, when you're trying to study the brain, lots of people object to having their brain removed for study. And so you have a much harder time of it, and you have to do the imaging at one step away. So the challenges are great. But the advances that are happening right now are really spectacular and are really opening doors.
Okay. Alan, I'm going to switch gears on you now that you've formulated your response to that one.
No, I'm both stressed and depressed now.
So despite the decades of research, doctors use symptom-based checklists to diagnose neurodegenerative disorders and mental illness. And we still don't have appropriate biological tests to confirm a diagnosis. How are we going to close that gap?
Right. The ultimate goal of what we're trying to do is to develop a tricorder. We have to come up with a tool that we can essentially stick it to your neck, and we can find out all of your biometrics and be able to incorporate that information into a more considered and informed characterization of your status. That's going to push us down the road of wearable technologies, all kinds of technologies to collect data that we've just been discussing. And the challenge for us is to capture all that information in a time which is compatible with clinical management. The doctor usually has to make a judgment call on 3 or 4 measurements that they have at their disposal with 40 people in the waiting room outside. So how can all of this information be made useful in a clinical context? I don't have an easy answer. It's not going to come overnight. We have to go through the research phase, if you will, of being able to collect all this data and extract the information from it using the kind of digital analytics that we've been discussing. Getting that information into the hands of the clinician is an extremely difficult thing to do. As I said, wearables gives us one time point. Genetic testing gives us another time point. Clinical measures gives us another. All of that we can capture, I think, in a reasonably rapid turnaround. That, I think, is close to being clinically applicable in the next decade. You're not going to put people in MRI scanners and PET scanners and all that fancy high-tech equipment in a time compatible with short-term clinical feedback, clinical management. So it's a glass half full over the next decade, I would say.
Okay, so this leads me right to the next question, which is, how has the understanding of the brain evolved in the last 15 to 20 years, and what gives you the most hope for the future?
Well, I think this characterization of the brain as a connectome is I think, revolutionizes how we think about the brain. We're no longer interested in associating some behavioral performance, some clinical assessment, with the size of the caudate nucleus or the size of the hippocampus. That is ancient history. We're very much interested in how different parts of the brain talk to each other in a circuitry context. Now that circuitry can operate at the macro level over the whole brain, different parts of the brain talking to different parts of the brain. It can also operate at the meso level, local circuitry. And you heard Rose talking about optogenetics and studying circuitry changes in response to some behavioral intervention. This is going to allow us to map the wiring of the brain at a level which was inconceivable just a few years ago. And that, I think, is completely revolutionizing how we think about the brain.
Okay, so in this younger set here, not saying that you're old, Alan, far from it, but in the younger, not the young, but the younger set, what do you look forward to? What excites you about what you think is 10 years down the line?
I'd like to say something that actually hinges on all of the questions at the same time. Something that I've noticed is that our expectations for the future for mechanical advances were grossly underestimated. But for understanding how the brain works, it was grossly overestimated. Right now, what I feel is that we have little mechanistic understanding of how the brain allows behavior differences to arise. I think that the path of network analysis is certainly one of the ways to go. But I do think that we'll eventually end up finding that it's even more complex and subtle than we could ever have imagined. So at this point, what I find promising is the huge computational power that is increasing year after year, and that might help us unlock some of these complex interconnections.
So I think it's on. I think what excites me most right now is seeing how boundaries are being broken down. And there's really this energy within neuroscience where all kinds of different people are coming to neuroscience and they're bringing their expertise from many different disciplines. And this is just really fueling this enormous energy and momentum that's revolutionizing neuroscience. So we have physicists, we have computer scientists, we have biologists, we have psychologists, we have chemists, we have data scientists. And I think that by breaking down these boundaries and having this sort of influx of new ideas and different abilities, we're really starting to approach sort of old questions in a fresh way. And I feel that we're starting to see these fundamental advances.
If I may as well, I'm just really excited to figure out what we got wrong. You know, in 10 years' time, what will we be looking back and thinking, gosh, why did we even think that? And I think some examples of that where we can look back now and think, oh, well, that was a little bit silly, was in the context of neurogenesis. You know, historically, we thought that we were born with a certain number of neurons and then they just died off along the lifespan and you had to be careful to keep what you had. But we now know that there's regions within the brain where there's new neurons being born every day, and that you can increase or decrease the amount of new neurons that are being born with different environmental, interventions. The same with regard to DNA. You know, we always said, well, your DNA is fixed and it's the same in every cell of your body, but we now know that there's mobile elements, transposable elements that can move around within the DNA, which means that the DNA in one cell could have subtle differences to the DNA in, say, for a skin cell, for example. So I think that's really exciting, not just for the sake of knowledge sake, but then understanding how these mechanisms play into pathological processes and then trying to use that information to ensure that we can use those processes and mechanisms to improve treatments and outcomes for people.
Well, I have to tell you, it makes me sorry I'm not younger, because this is going to be really interesting what's yet to come. So it's grand. Now, I have a question that in some ways I'd like to answer. So I'm going to ask myself this question. I could ask Suzanne the same question and she would answer it. So this is expensive research that you do. You have big equipment. You have lots of things that you need in your laboratories. You showed us a wonderful example. So how can society ensure that we cover the cost to equip our researchers? What needs to be done with the very best technology and training? And what role the private donors have to play through that philanthropy? So the answer I'm going to give you is what you really gave us, Mr. Ludmer, which is, this is expensive. We wanted to get a lot of money from the federal government in this contest of Canada Excellence Research Fund. And to do that, it was very good to have a leveraging effect, something that could help you show to the government, individuals, industries, people have a high degree of interest in this, such a high degree of interest that they're willing to put literally millions of dollars on the table. And that helps the wider world that's looking at these proposals to say, this is really important to people. People are really willing to back this. So just on the leveraging effect alone, this is enormous for us, an enormous role that philanthropy plays. So I'm willing to share the question with anybody else.
I'll weigh in on that with just one thought. I think that as scientists, we spend a lot of our time writing grants to fund our research, and that will always be our bread and butter. But by the same token, that research in some sense will often be bread and butter. It'll be a bit safe. But when we have private donors who have vision, we can do really exciting research because they have the vision to support the things that might be a bit riskier, but it's the risky ideas that have the biggest rewards.
Now I'm going to ask one last question. In research and academic circles, we often talk of taking research from the bench to the bedside. How can we ensure that the kinds of discoveries you're making end up transforming themselves, translating themselves, transferring themselves into better care and treatment for people?
Well, if I may, you know, I went to Washington a couple of years ago and was speaking to a federal agency on children's and families, and I was talking about some preliminary results that we had in an early intervention, and the woman was so excited because she was saying that, you know, if we can show that this has a biological effect, it will be easier for us to make our case that we need funding for these interventions. You know, by being able to understand biological mechanisms and the efficacy of different programs at a biological level, it actually makes it easier to fund these programs that we know can make a difference in people's lives. So I think biology has an important role to play in shoring up these programs to show how and why they work. And that leads to us understanding how we can make them work for more people than for specific people.
Okay, this is going to lead us into the next piece of this program, which is we have two people here that work really in the serving of the people end of this. We have somebody you've already met, which is Dr. Greenwood from the Jewish General Hospital, but we also have Neja Hashimi Idrisi, who I think is here. There you are, good. Okay, so I'm going to thank you as panelists, and then I'm going to invite Dr. Greenwood and the other contributor who has been recently appointed as the Assistant President and Chief Executive Officer of the Integrated Centre for Health and Social Services on the West Island of Montreal to say a few words. So we'll clear off the stage. We'll leave you here, Celia, and then invite.
Au nom de Madame Lynn McVeigh, présidente directrice générale du Centre intégré universitaire de santé et de services sociaux de l'Ouest de l'île de Montréal, permettez-moi de remercier Madame Suzanne Fortier principale et chancelière de McGill Université, Monsieur David Heidemann, notre vice-principal Health Affairs and Dean of Faculty of Medicine, ainsi qu'évidemment notre donateur majeur, Monsieur Arvin Ludmer, pour cette invitation cordiale. I would particularly like to acknowledge the generous support to our researchers, which is helping them create an outstanding research program. Can big data help explain how our brains work? Voilà une thématique qui me tient particulièrement à cœur comme ex-directrice des programmes santé mentale et dépendance au niveau du CIUS de l'Ouest de l'île de Montréal et actuellement présidente directrice générale adjointe. I'd like to remind us that it has been said earlier, brain disorders impact 1 in 5 people. Un individu sur 5 adultes, adolescents ou enfants va être affecté par un trouble mental au cours de sa vie. Et selon l'Organisation mondiale de la santé, ça a été mentionné également, la dépression sera bientôt la maladie numéro 1 en 2030. Also, 70% of mental illnesses are believed to have their genesis in the prenatal and early childhood. We heard about it also. Et aussi, malgré tous les efforts au niveau de la stigmatisation qui s'amenuisent quand même, le dialogue est de plus en plus ouvert, plus que jamais. Les moyens pour traiter les troubles mentaux sont encore rares et on en a témoigné également tout à l'heure. Et les innovations médicales aussi. So this means we need to act now to find therapeutic and preventive measures. Once relatively unknown field of research, epigenetics, is now understood to be a key mechanism in determining which genes are turned off or on. Mr. Michael Meany, a senior researcher at the Douglas Mental Health Institute and co-director at the Ludmer Centre, and his team now seek to develop a detailed, multi-dimensional picture of what vulnerability to mental illnesses looks like and what interventions can help conditions before they take roots in adolescence and early childhood. And big data analytics are of paramount importance in this research field. As a founding member of the Ludmer Centre Executive Committee, Mrs. McVeigh was instrumental in creating the first Ludmer Centre agreement and in establishing the Douglas Mental Health Institute as the Ludmer Centre's genomic and epigenetics pillar under the direction of Dr. Michael Meany. Today, we still believe that epigenetic research is crucial for the development of therapeutic treatments and can postpone onset or even avert an illness together, and to inform our ability to prevent mental illness from developing in the first place. So, as you all already know, the Ludmer Centre was established in 2013 as a partnership between the Neuro Institute, the Douglas Mental Health Institute, and the Jewish General Hospital Lady Davis Institute for Medical Research to improve prevention, diagnosis, and treatment of mental disorder through innovative research, as we heard earlier. Et aujourd'hui, plus encore demain, la mobilisation de nos forces scientifiques et la collaboration seront essentielles si nous voulons faire une différence dans la vie de nos patients en leur offrant des services adaptés et personnalisés. Et sur ces mots, je terminerai en vous souhaitant une bonne soirée et thank you all for your support.
Thank you very much. It's very important for us to have members of the community associated with our care facilities here and the research that goes on in them. Dr. Greenberg.
I'm just saying a few words on behalf of Dr. Rosenberg, who expresses his regrets that he could not be here tonight. He really wished he could be here tonight. He wanted to say that he's extremely happy to be part of the Ludmer Centre, to be one of the institutions that is part of the Ludmer Centre. And he's also extremely proud of the results that have been developed through the Ludmer Centre. And not only that, he looks forward to many exciting developments to come in the near future. So thank you.
And now it's my pleasure to invite Dr. David Eidelman, who is McGill's Vice Principal of Health Affairs and the Dean of the Faculty of Medicine, to say a few words. I think, or I've been told, he has something special to share with you.
Okay. I think I know what you mean, but that wasn't what I had my own plans. Alors, merci, bonsoir à tous. Merci. C'est un grand plaisir pour moi de pouvoir prononcer quelques mots à la fin de cet événement exceptionnel. Bienvenue. I want to particularly acknowledge Mr. Ludmer, and although it's not in my notes, I want to say that Mr. Ludmer was one of the first philanthropists that I met when I became dean. My former colleague, my predecessor, Dr. Fuchs, took us out for dinner. You may remember this, Irving. And I have to say that I learned a lot from Mr. Ludmer. I learned the value of persistence, even in the face of disaster. Despite our best efforts at not accepting Mr. Ludmer's money, he persisted and made sure we accepted it. And he did so because of his extraordinary vision that you've been hearing about tonight. And I really think it is really thanks to his determination that we're here. And I think that applies on all levels, scientific and other levels as well. Of course, I also want to thank and congratulate our panelists, Alan, Celia, Kieran, Rosemary, and Sharif. Those were really excellent presentations, whether they were 5 or 6 minutes long. I want to acknowledge our healthcare partners, as Martha was just doing. Obviously, this is a project, a McGill project, but it's also a project at the Douglas, at the Jewish, and of course at our own MNI. We're really fortunate in our community to have such extraordinary expertise available to apply to these really, really fundamental problems. Everybody's talked about the extraordinary opportunity of being able to bring people together. For me, in addition to the fact that this has the potential to really benefit patients and their families, it's also really cool. And I think that actually counts as an important factor in why we should do these things. So, j'aimerais maintenant attirer votre attention vers l'écran où nous présenterons une petite vidéo. Alors ça, je crois que c'est la surprise dont on parlait tout à l'heure.
Science is all about ideas and ideas that can change the world. And I think there's no better investment that a donor can make, really, than in the young generation of researchers that are coming up with some of the best and brightest ideas of our generation.
In my team, we currently have 4 graduate students, and they really drive the research. So they're in here every day doing the experiments to make the breakthroughs to drive the science forward. I actually did my PhD at McGill. I recall applying for one particular fellowship. Getting that award was such a pivotal moment for me in my career. I saw that I was competitive for funding, and that paved the way towards me becoming an independent researcher and an assistant professor at McGill.
Funding played a huge role in my career so far. I received a huge scholarship that allowed me to go to Stockholm to do my final year of undergrad, my honors thesis in a research lab over there.
These kind of funding awards enable students to be able to get the training that they need in various different skills, so they can just think, where do I want to go, and then, achieve their dreams for their research goals.
So, as you saw in the video, we're launching the new Ludmer Centre Student Awards for Transdisciplinary Brain Research, together with a crowdfunding campaign, which is also cool, to support students' work in mental health. And really, as several of the investigators said, there's really nothing more productive in terms of moving research forward than promoting our students, who are the people on the line who really do the work. These students work on a range of disorders, everything from autism and bipolar disorder to depression and schizophrenia. on to neurodegenerative diseases like Alzheimer's and Parkinson's. And tomorrow you're going to receive a link by email that will lead you to the crowdfunding campaign created specially for these awards so that you can contribute should you wish to do so. I thank you all for considering doing that, and merci beaucoup de votre présence avec nous ce soir et de votre contribution à la solution en santé mentale. Merci beaucoup et bonne soirée à tous. Thank you very much.